Usually when we talk about a medical diagnosis, there's this expectation of precision. It feels almost like engineering.
Right.
Like you break your arm, the X-ray shows that jagged white line, and the doctor just points and says, there it is.
Yeah, it's binary, broken or not broken. It's clean. And fundamentally, it's comforting. We like things to be visible and categorized.
Exactly. We want a clear cause and effect. But then you step into the world of artificial intelligence in healthcare, and suddenly that reassuring X-ray machine feels like it's, I don't know, printing out modern art.
It really does. It becomes this murky black box.
Right. An AI tells a doctor a patient has a high risk of heart failure, but it can't always point to the jagged white line. Well, today we're going to clear that up for you. Welcome to the deep dive.
Glad to be here.
We have a massive mission today. We're going to prove that the black box of medical AI is finally starting to turn translucent.
And you really need to care about this shift. Medical AI is moving way past simple image recognition now.
Oh, absolutely.
The research we're exploring today represents the absolute frontier. We are talking about systems that can reason through time, remember patient histories, and basically understand biological mechanics, not just match pixel patterns.
Exactly. And to pull this off, we are going to synthesize an entire stack of exactly 23 cutting-edge medical AI research papers.
That is a lot of ground to cover.
It is. And because we respect your time, we've got a really tight, strict window to hit our audio overview target of exactly 15 to 17 minutes.
We are definitely on the clock.
Right. So, to keep things rigorously organized without losing the thread, we will explicitly announce the paper number right before we dive into its specific breakthrough. Let's unpack this.
That's good. Where do we start?
Well, before AI can truly revolutionize medicine, we have to actually trust its outputs. And trust starts with knowing why an AI said what it said. That brings us to paper number 1 by 𝗂𝗍𝗍 and colleagues.
Yeah, this is a perfect starting point. When a large language model makes a medical prediction, tracking that prediction back to its original training data is notoriously difficult.
Because the tokens are so messy.
Exactly. The pieces of words it processes influence each other in these incredibly messy, overlapping ways.
Right, so paper number 1 solves this using something called orthogonal latent spaces. Basically, it's like finding the exact sentence in a massive medical textbook that caused a med student to misdiagnose a patient.
Instead of just knowing which library they studied in.
Exactly.
And what's fascinating here is how they achieve that precision. They use something called sparse autoencoders. Think of these as a filter.
Okay.
It disentangles all those messy token influences into clear, independent features. And once they have those, they use a mathematical tool called Jacobian vector products.
Which sounds intense.
It is a bit, yeah. But without getting bogged down in the calculus, it's just a way of calculating the exact sensitivity of the AI's final output based on one specific feature in the training data.
So it gives us token-level precision for AI accountability. That's huge. But tracing the data only helps if the data itself is complete, right? What happens when the model is forced to guess? Let's talk about paper number 12 by David Ageroux and his team. They looked at AI hallucinations in inverse problems.
Yes, like reconstructing an MRI image from raw, undersampled signals. A lot of people think of AI hallucinations as, software bugs.
Like a glitch that can just be patched.
Exactly. But this paper proves that in inverse problems, hallucinations are fundamental limits of the math itself.
Wait, really?
Yeah. If the raw data is physically incomplete, the math literally has multiple possible solutions. The model has to guess to fill in the blanks.
Wow. Which means we are going to have failures, and we need much better ways to test these systems before they hit the clinic.
Absolutely.
So how do we build a better stress test? That's the focus of paper number 2 by 𝗏𝗏𝗡 and team on counterfactual stress testing. Now, I have to push back here for a second.
Go for it.
Aren't we already stress testing these AI models? We mess with image brightness, we add digital noise, we rotate the scans. Why isn't that enough?
Because turning down the brightness on a screen doesn't mimic a real-world clinical shift.
Oh, I see.
Simple visual tweaks fail to capture what actually causes AI to break in a hospital. This paper introduces causal generative models to create realistic what-if scenarios.
What does a causal generative model actually do differently, though?
Instead of just applying an Instagram filter to an X-ray, it builds a causal mathematical graph of the patient's anatomy. It understands the physical structures independent of the scanner.
So it knows what the liver looks like regardless of the machine.
Right, so it can digitally change a patient's sex or simulate what their exact anatomy would look like if scanned on a completely different machine from a different manufacturer.
While preserving their unique anatomical identity, that's brilliant. It's a much more robust proxy for real-world out-of-distribution performance.
Exactly.
Okay, so trusting the model's reliability is step 1. But step 2 is fundamentally changing how the model interprets those images in the first place.
Yeah, the vision aspect.
Right. For years, we've trained models to look at scans and just highlight the problem areas. But paper number 3 by Siddharth Gupta and colleagues argues we need to move beyond simple per voxel masks.
Towards medical image parsing.
Right. And for you listening, a voxel is basically a 3D pixel.
And for a long time, segmentation models were just taught to color in those 3D pixels. Like, here's a red blob for a tumor, a blue blob for an organ.
Exactly. Segmentation taught models to measure. Parsing asks them to explain. Because a real radiologist's report isn't just a colored picture, right?
No, it contains spatial relationships, severity grades, clinical attributes.
The tumor is adjacent to the vessel, the margin is irregular, the AI needs to outquint that level of semantic relationships.
And that need for deep relational explanation connects perfectly to paper number 20 by Chen-Yu and team. They introduced a framework called EvaScreen.
Which is really cool.
It is. Historically, to explain an AI's decision, developers used post-hoc saliency maps. Those are just heat maps highlighting which pixels the AI looked at most.
But that doesn't tell a doctor why those pixels matter.
Right, so EvaScreen uses evidential reasoning instead. It pulls region-level evidence from dual knowledge banks of historical cases.
So it's not just highlighting pixels. It essentially says, I think this is a malignant lesion, and here are 3 structurally identical historical cases from our database with confirmed biopsies to prove it.
Precisely. It argues its case with precedents, like a lawyer. It anchors its perception in historical evidence.
But what happens when you encounter a completely novel anomaly? You can't always rely on past data to spot a new problem.
True.
That is the fascinating leap made in paper number 11 by Qiaowan and team on 𝗵𝗽𝗶𝗴𝗵𝗵. They tackle this using sparse hypomatching.
This is one that's like a biological immune system, right?
Exactly. Your immune system doesn't need to have seen a specific synthetic toxin before to know it doesn't belong in your bloodstream. It just recognizes that the structure is fundamentally alien to your healthy baseline.
And mathematically, they achieve this using hypergraphs. In a standard graph, an edge connects 2 points like 2 related pixels. But in a hypergraph, a single edge can connect multiple points simultaneously.
Capturing incredibly complex, multi-way relationships.
Yeah, it allows the AI to spot anomalies without prior training or explicit text prompts using purely visual hypergraph evidence.
That's amazing. But an immune system response requires looking at the whole picture.
Yeah.
In the real clinical world, you rarely get a perfect, uniform set of scans for every patient.
Oh, almost never. Which is the massive hurdle addressed in paper number 6 by Qiaowan and colleagues called Brain Anytime.
Because missing data is a huge problem.
Massive.
Yeah.
Often, a patient might only have a T1 MRI scan, while another has a full PET scan. Standard AI models just break if you change the input modalities.
But Brain Anytime accepts whatever arbitrary imaging is available.
Right. It manages this by learning structural molecular correspondences during pre-training. It learns how different scans correlate.
So it can logically fill in the conceptual blanks even when data is missing. All right, so we've solved how AI interprets static images, whether they are complete or missing parts. But patients aren't static. The next massive frontier is understanding how a human body changes over time.
Time is the hardest variable.
It really is. Let's look at paper number 21 by 𝗵𝗽𝗶𝗶𝗵 and team, evaluating the CHECKS temporal dataset. They looked at temporally grounded reasoning in chest x-rays.
And it turns out AI models are actually pretty good at seeing when a condition gets noticeably worse.
Right, because a massive new consolidation in the lung is visually obvious.
Yeah, but the models are terrible at spotting subtle longitudinal states like stable or resolved.
Which is so critical. To declare a patient stable, the AI has to hold a highly detailed baseline state in its memory and compare microscopic changes over a period of months.
And to fix that, we have to force the AI to actually think in steps.
Yeah.
Let's move to paper number 15 by 𝗪𝗁 and team on rad thinking.
Ah, right.
This is a visual question answering data set for cancer screening that forces multi-step chain of thought reasoning.
It forces the AI to move from atomic perception, like simply observing, I see a 2-centimeter lesion, to actually applying strict clinical guidelines.
It has to deduce step by step.
Yeah.
Because it is this size and has this type of arterial enhancement, it classifies as a LI-RADS5 liver tumor.
So it bridges the gap between seeing and deducing. But applying logic over time becomes infinitely harder when the timeline is moving fast, like in an intensive care unit.
The ICU is a nightmare for these models.
Which brings us to paper number 7 by Qiaowan and colleagues on Clin-JEPA. Now, I have a question here.
Sure.
If we want to track patient trajectories in the ICU, why not just use a standard autoregressive LLM? We use them to predict the next word in a sentence. Why not just predict the next medical event?
This raises an important point. And the answer is something called representation collapse.
Okay, what's that?
If you try to predict complex, high-dimensional medical events over a 48-hour horizon using a standard LLM, every tiny error compounds. The model's predictions quickly drift completely away from reality. Clin-JEPA solves this by using a joint embedding predictive architecture.
Meaning, instead of trying to predict the exact data points of the future, it predicts the abstract representations of the future.
Exactly. By co-training the encoder and the predictor simultaneously, without freezing them, it keeps the model firmly grounded in clinical reality.
That makes a lot of sense. And keeping track of that reality involves reading incredibly dense electronic health records, or EHRs. That's the focus of paper number 5 by Ming-Cheng Zhu and team with MedTPE.
EHRs are endlessly long. You can easily max out an AI's context window.
Right, so this method acts like medical shorthand for AI.
It compresses token sequences losslessly by up to 31%.
And the losslessly part is crucial, right?
Oh, absolutely.
Yeah.
You can't just summarize a medical record and accidentally drop the word "not" from "patient is not allergic to penicillin."
That would be disastrous. So MedTPE works by merging frequently co-occurring medical token pairs into single composite tokens, making the AI's processing incredibly efficient.
Spot on.
So we have an AI that can efficiently read a timeline and logically track a patient's state. The next evolution is for the AI to start acting within that timeline. We are entering the era of autonomous medical agents.
This is where things get really wild.
Let's jump into paper number 13 by Yuhao Wang and team. They introduce MedMemoryBench, which evaluates agent memory over long-term clinical tracking.
Here's where it gets really fascinating. They uncovered a major flaw in these agents called memory saturation.
Yeah, this blew my mind. You would think more data makes the AI smarter.
You'd think so.
But if you feed the agent too much sustained historical information over a long period, it actively degrades its reasoning.
The AI's attention mechanism literally gets diluted by the sheer volume of irrelevant historical noise.
Causing it to fail at connecting the dots for current acute problems.
Exactly. Which means if we are going to unleash these agents, they need safe environments to practice in to learn what information actually matters.
Enter paper number 8 by Minghao Wu and colleagues on sepsis agent.
Managing sepsis in the ICU is incredibly difficult. It requires constant adjustments to fluids and vasopressors.
Right, and this paper tackles it by building a clinical world model. It's essentially a flight simulator for AI doctors.
It simulates exactly how a patient's blood pressure and organ function will respond to a treatment before the AI actually prescribes it.
So it uses a proposed simulate refine workflow, safely learning the patient's biological dynamics in a virtual sandbox.
And we are taking that concept of biological simulation all the way down to the microscopic level with paper number 4 by Edward de Brouwer and team.
That's AssayBench, right?
Yes. This research tests LLMs on virtual cells to predict the phenotypic effects of cellular perturbations.
Meaning if you introduce a specific chemical compound to a cell, the AI can simulate how that cell's expression will change.
Right.
So if we can simulate the macro environment of an ICU and the microenvironment of a single cell, what about the physical mechanics of the human body itself?
How does AI interpret a body in motion?
Exactly. Let's announce paper number 22 by Jared Levy and team on dance.
They are moving away from clean, controlled lab data to real-world, continuous EEG monitoring for seizures.
And the major breakthrough here is asynchronous detection. In the real world, you don't know exactly when a seizure starts.
Right. There's no starting bell. Their pipeline can continuously analyze brain waves and detect events without known onsets, which is a massive leap for wearable monitoring.
And translating those external signals to internal biological states is exactly what paper number 17 by Yujun Hu and colleagues achieves with BioHuman.
This model looks at monocular video, just standard 2D video of a person moving, and predicts their internal muscle activations.
How does it even do that from a 2D video?
It uses inverse kinematics. It looks at the resulting motion and calculates backward to find the cause.
It's like looking at a complex puppet and being able to mathematically calculate exactly which invisible strings are being pulled and with how much force, just by watching how the puppet's arms swing.
That's a great analogy. The AI infers the exact biomechanical muscle firing underneath the surface.
And while we're analyzing movement, let's look at paper number 9 by Arafat Rahman and team on ESVAE.
Right. They analyze skeletal gait cycles, like tracking the exact mechanics of how stroke patients walk to monitor their recovery.
But the problem with video analysis is that a camera angle can distort the movement, right?
Exactly. So ESVAE solves this by mathematically stripping away nuisance factors like the camera viewpoint or the speed at which the patient is walking.
Leaving just the movement itself.
Yeah. It uses something called a shape manifold to focus strictly on the intrinsic geometric mechanics of the movement. It isolates pure, objective biomechanics.
So from the macro mechanics of muscles and bones, our deep dive now shrinks all the way down to the fundamental building blocks of those systems' proteins and molecules.
The molecular frontier.
Let's explore it, starting with paper number 10 by Juan Park and colleagues on MolDeTox.
This paper addresses a massive, often ignored issue in AI drug discovery. We spent so much time asking AI to find molecules that effectively bind to disease targets.
But we haven't focused enough on whether those molecules will actually poison the patient.
Right. MolDeTox evaluates stepwise fragment editing to systematically detoxify molecules.
Making sure we aren't just generating effective drugs, but safe ones. Which naturally brings us to paper number 18 by Qinshuan Zhang and team on Forge.
Yes.
Now, wait a minute. We use natural language prompts for everything these days. If I want an LLM to write code, I prompt it. Why can't we just prompt an LLM to design a better, safer molecule?
I get why you'd think that, but relying on plain text natural language for chemistry introduces a massive data scaling bottleneck.
Why is that?
LLMs are trained on human language, which is inherently ambiguous. In chemistry, ambiguity is lethal. It leads to what we call chemical hallucinations.
Oh, wow.
Yeah, an LLM might string together a sequence of molecular symbols that sounds highly plausible in text, but utterly violates the physical laws of chemistry and reality.
Okay, so Forge fixes this through explicit fragment-level supervision. Instead of relying on expensive human text annotations, it breaks molecules down into chemical fragments.
Right. It ranks candidate fragments by their actual property contribution and generates explicit structural replacements.
Bypassing the language hallucination entirely.
Precisely. It anchors the AI in chemical reality. But optimizing a molecule is a multi-step journey, which brings us to paper number 19 by Haoji Rao and colleagues on 𝗪𝗏𝗏.
Right. This paper solves the mismatch between our high-level property goals like make this drug more soluble and the strict single-step chemical feasibility rules.
And it does this without relying on an unstable oracle-in-the-loop search.
Right, because when an AI has to constantly pause and ask an external chemistry oracle simulator if its next step is valid, it slows the entire optimization process to a crawl.
𝗪𝗏𝗏 plans multi-step molecular optimization far more efficiently.
But here's the catch. When we start fine-tuning these models to align with highly specific medical preferences, we run into a major cognitive roadblock. Let's bring up paper number 14 by Yulin Zhang and team on protein OPD.
This is a wild concept. When you fine-tune an AI heavily toward a specific goal, say, designing a protein that binds perfectly to a novel virus, the model often suffers from catastrophic forgetting.
It gets so hyper-focused on the new goal that it literally forgets the basic thermodynamic rules of protein designability. It loses its foundation.
To prevent that tunnel vision, protein OPD uses on-policy distillation. Think of it as a mathematical safety net.
Okay.
While the model is updating its weights to learn the new preference, the distillation process constantly pulls it back toward its original fundamental knowledge base.
So it balances learning the new specific task while strictly maintaining its grasp on the basic laws of physics.
Exactly.
And if we want those perfectly designed proteins to actually interact with other biological systems, we need paper number 16 by Ziki Gao and team on L3PPI.
Historically, AI tried to predict protein interactions using generic math, like simple dot products.
Which ignores the biological reality of how these systems work.
Right, so this model bakes in the biological L3 rule via graph prompt learning.
And the L3 principle in network biology basically states that proteins don't always interact directly, right? Their interaction relies heavily on shared intermediate partners.
Yes. By using graph prompt learning, the model literally encodes that specific biological geometry into the AI's logic. It's using the actual rules of biology to teach the AI rather than just throwing brute force math at the problem.
Which brings us perfectly to our final piece of research. Let's introduce paper number 23 by Andrew Y. Zhou and colleagues on ToolMol. This is an evolutionary agentic framework for multi-objective drug design.
And it is the perfect capstone for everything we've talked about today.
Because instead of letting the LLM act like a black box and randomly guess letters in a molecular string, ToolMol gives the agent tools.
Right. It equips the AI with an exact, already kit-backed chemical scalpel.
When the AI wants to modify a molecule, it has to use a validated software tool to make a precise, chemically valid modification.
It completely revolutionizes the process. It combines the broad, creative reasoning of a large language model with the strict mathematical precision of an evolutionary genetic algorithm,
resulting in highly synthesized, profoundly safe ligands with incredibly strong binding affinity.
Exactly.
And with that, take a deep breath. We did it. We have successfully navigated an extraordinary journey, moving from pixel-level interpretability and AI accountability through the simulation of patient timelines and physical motion, all the way down to agentic evolutionary molecular design.
We really covered it all.
We explicitly called out the breakthroughs across all 23 papers, and looking at the clock, we are perfectly landing within our 15 to 17-minute audio overview window. mission accomplished.
It really is a remarkable stack of research to synthesize. If there is one core takeaway for you today, it's this AI in healthcare is no longer just a passive tool for identifying tumors on a screen.
Definitely not.
It is rapidly evolving into accountable, reasoning entities that understand the flow of time, the biomechanics of physical human motion, and the strict physical constraints of chemistry.
It's breathtaking how fast the frontier is moving. But I want to leave you with a final thought to ponder today.
Let's hear it.
If an AI system can simulate a virtual cell's reaction to a drug, if it can propose and refine life-saving treatments in a simulated ICU world model, and if it can flawlessly track a lifetime of complex medical memory...
Oh, wait, no ellipses.
Right. Strike that. Let me say it like this. At what point does the AI become the primary physician and the human doctor becomes the co-pilot, simply verifying its work?
It's a profound shift in the medical paradigm.
Truly. Remember at the start of our deep dive, we talked about that comforting precision of a broken bone on an X-ray versus the murky black box of AI.
Yeah.
Well, looking at where these models are heading, explaining their reasoning step by step, simulating our biology, and mapping the exact chemical scalpels they use, it seems that black box is finally turning translucent. The jagged white line of truth is coming back into focus. Thanks for joining us. Keep learning.